Hi everyone, welcome back to the channel. Dr. Rassi Explains. This is Dr. Rassi, and today's topic is the lab exam 2, BSC1010 lab exam 2 study video. And let's get started. So in this module 2, we have 3 labs: cell membranes, enzymes, and photosynthesis. So 3 labs you need to study. So we start with the first lab, which is cell membrane or membranes. So in this lab, we actually cover membranes, and we did experiments to test membrane transport. So always remember, membranes, it could have passive transport like diffusion, like solids go from high to low, or it could be osmosis, which water will go from high water to the low water. The same idea, but they're both passive transport, right? So the experiment we tested that was a agar media inside a petri dish, and then we put 2 different dyes, and then we see which dye actually is gonna move faster. So that was typical diffusion. And active transport will be any direction, even uphill or downhill, which the only difference is it will require energy or ATP, which is not free. So we also did experiments on cell and its environment, like if it is hypertonic, if you put a cell inside a salt concentration. So obviously that's a very hypertonic situation. And then 2 things is gonna happen. Water from inside cell is higher, it's gonna move out, and cell is gonna shrink, and because this is typical hypertonic, until it becomes same in both levels, which is what's called isotonic. The opposite this will be if something called hypotonic, which outside is more water and inside is less water. So you are going to test this with, you already tested this with an experiment with Elodia leaf, and then put them on salt and water, 2 different slides, and then see which one shrinking, which one is not shrinking. So, which is also called plasmolyzed if it loses water and shrinks. Another thing we did in this lab was actually serial dilution. So serial dilution is you always have a stock solution, and you need to make lower concentrations from that. You can go highest is your stock, maybe 5% salt. And if you want to go lower concentration of this salt, with one technique is called serial dilution, which is basically we use 6 test tubes, and then 1, 2, 3, 4, 5, 6. We put water in 2, 3, 4, 5, 6. Then we didn't touch 6 anymore. That's our negative control, just with water. We put 4 milliliter salt concentration in one, and we took 2 from 1 to put 2, took 2 from 2 to put 3, 2 from 3 to put 4, 2 from 4 to put 5, and then took 2 from 5 and throw away. Now we have this series of concentration. We basically we halved each every time. That means we really twofold. We diluted this each. So if we start with 5%, we end up 2.5, 1.25, 0.065, 0.3, and 0, which is 6 test tube number. Last test tube, which is test tube 6. You can also use a formula I mentioned in the lab, C1 times V1 equals C2 times V2. You can also use that to do the same thing. This was lab number 5, which is basically enzymes. So membrane. This is lab number 6, which is enzymes, the next lab. So remember, in enzyme, we also did a serial dilution. The idea is exactly the same. So to dilute the enzyme, our enzyme here is amylase. It's also called salivary amylase. Every enzyme has a substrate. our substrate for this enzyme is starch. If you combine them, they actually are going to break down this starch into the maltose and glucose. And we can measure this by using Benedict solution. Our color change is positive color change is orange, right? But we need to heat these test tubes to actually see this color change. So we tried 3 different experiments, optimal concentration, optimal temperature, optimal pH. So look at your results. Remember, this is an enzyme in the mouth, which is saliva. And so because of that, our expected optimal temperature will be around 37 degrees because that's body temperature. Optimal pH, again, around 7. That's neutral pH. So just, and then serial dilation, same idea, but this time serially dilating enzyme. That was lab number 6. And then lab number 7 is basically photosynthesis, right? So lab number 7 is photosynthesis, and we did a number of experiments testing photosynthesis. Remember the photosynthesis formula. We can measure a number of things in that formula, including CO2, oxygen, chlorophyll, light, and some others. So photosynthesis is made of light reactions, dark reactions. Light must exist in the light reactions. We can test this using actually a spectrophotometer, basically to test the light, how much light absorbed by our leaves. We use 3 different plants. You can see in this picture, the coleus, spinach, geranium, and also elodea, 4 different plants, and to measure different things. Light, again, to measure light, we use spectrophotometer. To measure CO2, we use a dye called phenol red. It's usually red color, but if you actually blow air to it, that means we're blowing CO2 into it, it will turn yellow. If you put a plant and put it in a light, it will remove the CO2, it will go back to the red. So it's called phenol red. Another experiment was testing white light versus dark. That's using geranium from a dark cabinet and a light geranium, and then test them. We basically boil them in water and ethanol, and then took them, put some iodine on them, and look at the color change. Again, color change here is black, is the correct color. And then for chromatography, we use spinach plants, and then we crush them and we get their solution, green solution, and then we use that to put a few drops on a chromatography paper, and then rank them, wait until they run from lowest to highest, from chlorophyll B, chlorophyll A, xanthophyll, and all these colors. So make sure that check that in the lab, and then make sure that you know the correct color. Again, this is in photosynthesis experiment, which is the last experiment in module 2. So the correct colors are carotene at the very top, like orange color, xanthophyll, yellow, brown, chlorophyll A, blue, green, and chlorophyll B, yellow, green. That's the correct order. So these are the 3 labs, and please review your notes and always study your book, the orange book, BSC1010 lab manual. Best of success in the lab exam too. Thank you for watching, and please feel free to like and subscribe our channel, Dr. Asi Explains. Bye now.